day, sir. My name is Princess Gail Perez Salonsan, a third-year criminology student of Philippine Countryville College, taking up the course of Bachelor of Science in Criminology. Before anything else, I am here to discuss to you the blood and blood stains. What is blood? Blood is a constantly circulating fluid providing the body with nutrition, oxygen, and waste removal. Blood is mostly liquid, with numerous cells and proteins suspended in it, making blood thicker than pure water. The average person has about 5 liters, more than a gallon, of blood. Moreover, detailed analysis of the blood obtained from a stain can reveal genetic and other information that can help identify a victim or implicate the person responsible. What is the importance of blood and blood stains to a crime investigation? Properly collected and preserved blood evidence can establish a strong link between an individual and a criminal act. The composition of blood. The composition of blood has 4 main components, which are the plasma, the red blood cells, white blood cells, and platelets. Blood has many different functions, including transporting oxygen and nutrients to the lungs and tissues, forming blood clots to prevent excess blood loss. Plasma. Plasma is the largest part of your body. The main role of plasma is to take nutrients, hormones, and proteins to the parts of the body that need it. Cells also put the waste products into the plasma. Red blood cells. Red blood cells is a type of blood cell that is made in the bone marrow and found in the blood. Red blood cells contain a protein called hemoglobin, which carries oxygen from the lungs to all parts of the body. It may be used to look for conditions such as anemia, dehydration, malnutrition, and leukemia. White blood cells. White blood cells are part of the body's immune system. They help the body fight infection and other diseases. Types of white blood cells are granulocytes, neutrophils, eosinophils, and basophils, monocytes and lymphocytes, T cells and B cells. Blood platelets. Platelets or thrombocytes are small, colorless cell fragments in our blood that form clots and stop or prevent bleeding. Platelets are made in our bone marrow, the sponge-like tissue inside our bones. The chronological test for blood. Preliminary test. Preliminary test, also known as screening test or field test. It establishes the possibility that a specific bodily fluid is present, do not conclusively prove the presence of a specific substance. Confirmatory test. Confirmatory tests for blood include identification of blood cells under a microscope. Crystal tests such as the Teichmann and Takayama test. Precipitin test is also known as biological reaction for proteins. It enables us to distinguish between proteins by using the animal as a test tube and to establish differences between them which no other form of test tube will detect. Blood grouping test. The test to determine your blood group is called ABO typing. The sample is checked to see whether or not the blood cells stick together. If blood cells stick together, it means the blood reacted with one of the antibodies. Preliminary test for blood. The benzidine test is most commonly used for detection of blood and was therefore singled out for investigation. Phenolphthalein test is a presumptive test that reacts with the heme molecule present in blood. A positive reaction gives a pink color, while blood stains normally appear red-brown in color. The color of the substance or the age of a stain may affect the appearance or visibility of the stain. Number 3 is the Guaiac-Combe test. The Guaiac fecal occult blood test, or the FOBT, is used to find occult blood, or blood that can't be seen with the naked eye. The idea behind this test is that vessels at the surface of larger polyps or cancers are often fragile and easily damaged by passing stool.
Hello. More than 145,000 people are diagnosed with colon cancer, and 57,000 people die from the disease. Men, women, and all racial and ethnic groups are affected by colorectal cancer. However, the disease is most common in people over age 50. The good news is that colon cancer can be prevented or even cured with early detection testing. The FOBT kit includes a set of 3 cards. Each card has 2 boxes, which are used to collect the stool sample over 3 days. There are applicator sticks that are used to collect the stool. And there is a special envelope to mail back the stool sample. To ensure the test accuracy, you need to follow your doctor's instructions. At least 3 days before collecting your stool sample, you need to change some of your diet and stop taking some medication until the test is over. Each time, before you collect your stool sample, you should fill out all the information on the front of each card. There are many methods to collect your stool sample, but it is very important that you collect it properly in order to avoid contamination. To collect your stool, you can put toilet paper in a clean container. Or another method would be to turn off your water supply to your toilet. Your water supply valve is usually located behind and below your toilet. Once you close your valve, flush your toilet once and there should be no more water left in the bowl. Now you can place some toilet paper inside the toilet. After you have your bowel movement, use the application stick to collect some of your stool. Then, smear a thin stool sample in the box labeled A. Then use the same stick to get a different sample of stool, and smear it in the box labeled B. Let the samples dry, and then close the card. After collecting the stool sample, be sure to wash your hands using soap and water. Turn on your water supply, and then flush the toilet. Continue to use the same method to collect the second and third stool sample until all the cards have samples. Remember that during this time period, you must maintain the food and diet restrictions until all the samples have been collected. Once all the cards have the samples, place them in your envelope. Then, send it to the laboratory or return them to your doctor's clinic as soon as possible. I hope these instructions have explained to you FOBT procedures. FOBT is the easiest way to detect colorectal cancer from your own home. Remember, colon cancer can be prevented or even cured with early detection testing. Thank you.
Number 4. The leukomalachite green test. The leukomalachite green test for blood is a catalytic test which is based on the peroxidase-like activity of hemoglobin. Hemoglobin has the ability to cleave oxygen molecules and catalyze the reaction from the reduced form of leukomalachite green to the oxidized blue-green colored product. 5 is the luminol test. The luminol or chemiluminescence reaction is responsible for the glow of lights. The reaction is used by criminalists to detect traces of blood at crime scenes. In this test, luminol powder is mixed with hydrogen peroxide and a hydroxide in a spray bottle. The confirmatory test for blood. The actual proof that a stain is blood consists of establishing the presence of the characteristic of blood pigment, hemoglobin, or one of its derivatives. Hemoglobin is the red coloring matter of the red blood cells of the blood. The confirmatory test has 3 whether stain is really blood: microscopic test, microchemical test, or the microcrystalline test, and the spectroscopic test. What is microscopic test? Microscopic test for a blood smear test, a laboratory professional examines the slide under a microscope and looks at the size, shape, and number of different types of blood cells. These include red blood cells, which carry oxygen from your lungs to the rest of your body. White blood cells, which fight infection. Microscopic examination, which identifies and counts the type of cells, casts, crystals, and other components such as bacteria and mucus that can be present in urine.
To flow through the smaller vessels and capillaries, they need to kind of fold over on themselves a little bit to fit through. So these biconcave discs are, in terms of their morphology, if we were to look at those closer, there's no detail that can be seen in here. These cells just carry hemoglobin. There's no nucleus, there's no organelles present, okay, which accounts for their sort of bland sort of appearance here. That's the red blood cells, which make up the biggest cell number. In amongst the red blood cells, which are the most numerous cell type in bloods, we can see some little dots which just look like bits of rubbish or extra bits, but these are platelets, and these are chips of cells which are in the bone marrow. These cells are called megakaryocytes, and platelets are simply portions of these cells that move throughout the blood and have organelles but no nucleus present, and these are not active in the normal situation within the blood, but are able to become activated and clot when the blood is breached, or in the case of thrombus formation in a vessel. A thrombus is a blood clot that's formed within a vessel, and this happens when the blood is exposed to tissue components, such as when the endothelium of a blood vessel is breached. If we have a look at the white blood cells, these are the ones that appear purple on this stain. Let's have a look at a few of them. There are 2 types of white blood cell. One is called the granulocyte series, and these are comprised of neutrophils, basophils, and eosinophils. And there are agranulocytes, which have no granules in their cytoplasm, and they are lymphocytes and monocytes. And rather unusually and luckily, we have in this field of view all 5 of those cell types. White blood cells make up only about 1% of the population of blood cells, and you can see that here represented. So there's lots and lots of red blood cells, but only very few white blood cells. So if we have a look at the most numerous cell type in the granulocyte series first, these are the neutrophils. Neutrophils are sometimes called polymorphonuclear leukocytes, or PMNs. That's because the nucleus has several different lobes to it, and so is a multiple shaped nucleus, or polymorph. That's one, that's another one, and there's one over here and one over here as well. They don't immediately look like they've got granules in their cytoplasm, although if you look at the cytoplasm it does look a little bit granular. And that's because the granules in there are neutrally staining. They don't stain very positively with either the hematoxylin or eosin component of the stain, and so they're neutral granules, hence the name neutrophil. Neutrophils are most heavily involved in acute inflammation. The other granulocyte we can see here is another cell with a lobed nucleus. In this case, the purple is the nucleus, and you can see one lobe here and a bit of a connecting bit and another lobe over here. And this is an eosinophil, and it has very dense pink, dark pink staining granules in the cytoplasm, and those pink granules are called eosinophilic because they're the eosin part of the H&E dye is attracted to them, giving them a very dark pink granular staining. And these cells are involved in parasitic infections and also involved in allergic reactions as well. If we have a look at the final white blood cell, this one is a basophil, and in this case there are very few of these in the blood. There's only about 1% of white blood cells that are basophils. This one is spottable as a basophil because it's got so many granules that the nucleus is completely obscured. The nucleus in this cell would be a lobed nucleus also, but it has, in this case, large purple granules that stain the same colour as the nucleus. Basophils are called basophils because they attract the same bit of the dye as the nucleus, which is hematoxylin, which is a basic dye, so they're called basophils. We now look at the last two cell types, the agranulocytes, which, as the name suggests, have no granules. In this case, we have the most common one, which is a lymphocyte. You can see here. A lymphocyte is a small cell. If you look at these other ones, they're a great deal bigger than a red blood cell, whereas the lymphocyte isn't really that much bigger than a red blood cell, and it's quite notable because of its nucleus takes up almost the entire cytoplasm, and it's just got a small rim or crescent of cytoplasm visible. That's how you identify a lymphocyte, and these are involved in immune reactions. There are many different types of lymphocytes, but we can't tell just by looking at them which is which. And the final cell type we have is the monocyte. It's quite a large cell and has a pale cytoplasm because there are no granules here, and it has a nucleus that is the shape of a kidney bean, and that is a monocyte. And monocytes are also involved in fighting infection. These are in small numbers in the blood. You find monocytes in tissue, and once the monocytes leave the blood they're called macrophages, and you find them in greater numbers in the tissue as macrophages than as monocytes in the blood. So in summary, the blood film that we're looking at here contains mostly red blood cells with a small number of white blood cells, which are
The microchemical test or microcrystalline test. Microcrystalline tests are chemical tests resulting in the formation of unique microcrystals for a given substance when combined with a specific reagent. Microcrystals are observed under a microscopic, and micrographs or microvideos constitute the results of the test. Spectroscopic test is described whereby the percentage oxygen saturation of hemoglobin within intact corpuscles can be determined. The microchemical test and microcrystalline test for blood can be made more specific if microchemical or microcrystalline test is applied or performed. The Takayama test and Takman test are the most popular ones. The three microchemical and microcrystalline test for blood. The Takman test, the acetone test, and the spectroscope test for blood. The Takman test is a confirmatory test for blood based on the formation of distinctive hematine crystals that are viewed under a microscope. The acetone test. A ketones in blood test measures the level of ketones in your blood. Ketones are substances that your body makes if your cells don't get enough glucose or blood sugar. Glucose is your body's main source of energy.
Testing for ketones is relevant for people with type 1 diabetes. In type 1 diabetes, a lack of insulin results in a higher level of ketones, which in turn can lead to a more dangerous state called ketoacidosis. Diabetes UK recommends that people with type 1 diabetes test for ketones if blood glucose levels become too high, usually above 15 millimoles per litre. Testing for ketones in this way could help you get advice and treatment before ketone levels become too dangerous. There are two different ways to test for ketones. The traditional way has been to use urine ketone testing strips. More recently though, some blood glucose meters have been developed that can test the blood for ketones. A blood test for ketones is similar to how you would do a blood glucose test. For more information on this, see our How to Do a Blood Test video. The following ketone testing advice is based on the recommended usage for Bayer keto sticks, like these. 1. Before testing, check the expiry date on the tub to ensure the strips are good for use. 2. Remove a strip from the tub, taking care not to touch the spongy end of the strip. 3. Pass urine over the test area of the strip, or alternatively, collect urine in a container and then dip the test area of the strip into the urine. 4. Begin timing immediately after applying urine. The strip will begin to change colour. 5. After 15 seconds, compare the colour of the test area to the colour chart on the side of the tub of strips. 6. Disregard any colour changes that might happen after 15 seconds has passed. Bayer provides the following advice for management of ketone levels. A level below 0.5 millimoles per litre. This is a trace level of ketones. No action is needed. around 1.5 millimoles per litre. This indicates that ketone levels are slightly higher than normal. It's recommended to drink a glass of water each hour and perform a follow-up test 3 to 4 hours later to see if the ketone level has decreased or not. If blood glucose and ketone levels do not decrease after 2 tests, contact your health team for advice. Around or above 3.0 millimoles per litre. This is a higher reading. Contact your health team immediately for advice.
Hemichromogen crystal test. Hemichromogen crystal formation with minute amounts of blood is described whereby small amounts of blood, such as the residual material from the precipitin test, may be used positively to indicate the presence of blood via the formation of pyridine hemochromogen crystals. The spectroscopic test for blood. A spectroscopic method is described whereby the percentage oxygen saturation of hemoglobin within intact corpuscles can be determined. Spectroscopy is a class of tests which study the interaction of sample specimens with energy, usually in the form of light. Heredity of blood groups. Just like eye or hair color, our blood type is inherited from our parents. Each biological parent donates one of 2 EBO genes to their child. The A and B genes are dominant and the O gene is recessive. There are 4 main blood groups or types of blood: type A, type AB, and the type O. Your blood group is determined by the genes you inherit from your parents. Blood group inheritance. Blood groups are inherited from both parents. The ABO blood type is controlled by a single gene. The ABO gene with 3 types of alleles inferred from classical genetics. Terms to ponder. Number 1. Gene. Gene is the basic physical and functional unit of heredity. Each consists of nucleotides arranged in a linear manner. Most genes code of a specific protein or segment of protein leading to a particular characteristics or functions. Number 2 is the chromosomes. Chromosomes are thread-like structures located inside the nucleus of animal and plant cells. Each chromosome is made of protein and a single molecule of deoxyribonucleic acid or the DNA. Chromosomes are the small bodies in the nucleus of a cell that carry the chemical instructions for reproduction of the cell. They consist of strands of DNA wrapped in a double helix around the core of proteins. Number 3 is the phenotype. The term phenotypes refers to the observable physical properties of an organism. These include the organism's appearance, development, and behavior. An organism's phenotype is determined by its genotype, which is the set of genes the organism carries, as well as by environmental influences upon these genes. Phenotype, in other words, is an individual's observable traits such as height, eye color, and blood type. Some traits are generally determined by the genotype, while other traits are largely determined by environmental factors. Number 4. Genotype. Genotype refers to the genetic makeup of an organism. In other words, it describes an organism's complete set of genes. Homogeneous genotype or pure genotype, the presence of two identical alleles at a particular gene locus. A homogeneous genotype may include two normal alleles or two alleles that have the same variant. Gamete. Gamete are an organism's reproductive cells. They are also referred to as sex cells. Female gametes are called ova or egg cells, and male gametes are called sperm. Sperm cell or microgamete refers to male sexual cell. Egg cell or microgamete, also defined as female sexual cell. Zygote. Zygote is a fertilized egg cell that results from the union of a female gamete, egg or ovum. In the embryonic development of humans and other animals, the zygote stage is brief and is followed by cleavage, when the single cell becomes divided into smaller cells. The alleles. An allele is a variant of a gene. Humans are called diploid organisms because they have two alleles at each genetic focus, with one allele inherited from each parent.